Okay guys, so today we're going to be talking about something a little bit different. We've been talking about DNA structure and replication over the past couple of days. So today we're going to be talking about something called RNA. It's three types: the structure and a process called transcription. So the last class that we talked about DNA in, we were able to make a copy of it, and there were three steps to that. We had DNA helicase that would break the hydrogen bonds between the double helix. Then we had DNA polymerase that would add DNA free-floating nucleotides to the strands, and then we ended up having two identical copies of DNA, and we're ready to divide. The cell was ready to divide and move on. So today we're going to do something similar but also different. Okay, so here we go. In order to make a protein, because that is our entire goal through this unit, is to take the instructions in DNA and make a protein. In order to do that, we have to use something a little bit different than just DNA. The reason for that is that DNA has to be kept locked up in the nucleus, otherwise it could get damaged or altered. So we're going to change DNA's message, its instructions, into something called RNA. So if you remember what DNA means, DNA is deoxyribonucleic acid. That would mean that RNA is ribonucleic acid. It is still a nucleic acid because it is found mostly in the nucleus. Its function, its job, is to take the DNA message or the instructions, its copy of the instructions, to be turned into, to be used to turn into a protein, to make a protein. And we're going to learn how to do this. If you notice here on this slide, there are a couple of things that might appear to be different to you looking at a piece of RNA. One of the first things that could stick out to you on the right-hand side here that I've kind of highlighted for you is that we now have a letter U with our bases now instead of a T. That's because RNA is not going to follow that GCAT rule like DNA does. It is going to have a brand new rule called GCAL or GCA, whichever way you're more comfortable with remembering it. The guanine and cytosine, the G's and C's, are still going to go together, but adenine is now going to pair with uracil instead of thymine, so no T's in RNA. Something else that you'll also notice is that it is RNA and not DNA. In DNA, we use the sugar deoxyribose. In RNA, we are going to use the sugar ribose. The last thing you might notice that is a little bit different here is that RNA only has one strand. So this is not a double helix. This is simply a helix. There are three types of RNA that we're going to be discussing. The first type is called messenger RNA, then we have transfer RNA, and we will also be discussing ribosomal RNA. Messenger RNA, for short, it's called mRNA. mRNA is only a single strand, so it's really boring. It's like one long piece of spaghetti. So nothing too exciting there, and it doesn't have any folds or clumps, and that's important to note because the next two RNAs that we talk about are a little unique. Sorry guys, it's like way past my bedtime. Okay, so mRNA is going to be found in the nucleus, and it's special because it's going to get to leave the nucleus, move out into the cytoplasm, and head for a place called a ribosome. So it is in the nucleus because its job is to rewrite the DNA instructions as RNA. Because remember, DNA can't get to a ribosome because it can't leave, but mRNA can. So it's going to take the DNA message, the instructions, to a ribosome. That is its function. So far you know its shape, its location, and its function. And also remember, it is going to follow that base pair, our new base pair rule of GCal. This is a very simple representation showing you the shape of mRNA. Like I said, it looks like a piece of cooked spaghetti. This is actually just yarn on paper, but I wanted to give you a better visual of its shape. The next type of RNA that we're going to be discussing is called transfer RNA, or for short, it's called tRNA. Now, if you look on the left here, it is called tRNA because it has this T shape, because it's kind of folded in that shape. It is only found floating around in the cytoplasm, and its job, its function, is to bring amino acids to a ribosome. If you think back to our cells unit, amino acids are the monomer, the individual Legos that build a protein. So this guy, the transfer RNA, is responsible for bringing every single Lego to the ribosome to get built into the protein castle. Okay, so if you look in the highlighted region here at the top of this tRNA, you see alanine. Alanine is one of many types of amino acids. This tRNA molecule just happens to be carrying the amino acid alanine. At the bottom in the highlighted region, there are 3 bases down there. You might not be able to see them too well. It's A, G, and C on this particular tRNA molecule. That is called the anticodon, and it's going to come in, have a really important role when we do notes tomorrow when I teach you a new skill. So knowing what's an anticodon and what a codon is going to be super important for tomorrow. The way I kind of remember this is I have a T shape that the amino acid is kind of like the hat on top of the tRNA. And you know me, I like to make you feel weird. The anticodon is kind of like the junk in the trunk on the bottom of that T. So again, that amino acid here is at the top. You have your T shape, and then you have your anticodon towards the bottom of that tRNA molecule. The last type of RNA molecule that we're going to be talking about is ribosomal RNA. This is also known as rRNA. The reason it is called ribosomal RNA is that this is a ribosome. A ribosome is made of ribosomal RNA. And it is just a big, messy glob clump of RNA. And the ribosome's job, again, thinking back to our cells unit, is to make a protein. So ribosomal RNA's job is to take that DNA message from mRNA and turn it into a protein. Again, that cells unit, ribosomes can either be floating around in the cytoplasm, or they can be attached to the endoplasmic reticulum, and that makes it bumpy, so we call that the rough ER. This is my sad attempt at making an rRNA model for you to just kind of get an idea of what's going on. But the question is, how do we get from DNA to RNA? The answer is a process called transcription, and this just means to rewrite DNA as RNA. And this is going to take place in the nucleus. The way to remember that this is going to take place in the nucleus is that if DNA is involved, it has to be in the nucleus, because DNA can't leave the nucleus. It could become damaged or broken if we were to allow it outside of the safety of the nucleus. So this is going to take place in the nucleus. DNA is going to be rewritten as RNA. Transcription is going to take place in 3 phases or steps that are very familiar to you. It looks a lot like DNA replication that we learned on Monday. So with DNA replication, you had an enzyme that broke hydrogen bonds holding the DNA double helix together, right? Same thing for the very first step of transcription. There's an enzyme, it's going to break those hydrogen bonds, so the DNA kind of unzips itself. And there's a reason I say unzips. So you have your DNA strand here on the left. You have an enzyme moving along that strand from the right to the left, and it's kind of unzipping. So you can kind of see how this looks like a zipper. So this looks very familiar to you. It looks just like DNA replication. The first steps in the 2 processes are identical. In the second step or phase, you are going to add RNA nucleotides to one strand of that DNA. So this is very much like that second step in DNA replication, but instead of adding DNA nucleotides, we're going to add RNA nucleotides. And we're not using the GCAT rule, we're using that GCAU rule where we have uracil instead of thymine. So let's take a look. So we have our double-stranded DNA that has been unzipped by the enzyme. The DNA is in yellow. We have RNA nucleotides being added to one strand of that yellow side. So at the top here where it says RNA nucleotides and the blue bars are pointing downward, you see that there is a G on the RNA. If there's a G on the RNA, what must be on the DNA then? It has to be a C. If there's an A on RNA, what goes with A on the DNA? A T. So this would go on for the entire strand until we've got everything copied that we need to have, or not copied, rewritten as we would want to be rewritten. So then we move to the third and final phase of transcription, and this is just when everybody calls it quits. The enzyme and the newly formed RNA molecule are released from DNA. DNA zips back up, and RNA can now leave the nucleus and head towards a ribosome to deliver the instructions for building a protein. Okay, so here you can see how DNA has been re-zipped, the enzyme has been released, and the mRNA has moved from the nucleus into the cytoplasm. So for a little bit of practice, I would like you to take out a sheet of paper. In the middle of your paper, you're gonna at the top, you're gonna write DNA, and then extending downward this column of GCAT bases that we have here. Then on the left side, you will have a new column of DNA. So if I have DNA and I'm making more DNA, this is the process of replication. So because this is DNA, you are going to be using the base pair rule of GCAT. So I'd like you to take a moment to construct this side of the DNA molecule. If there is an A here, what would be on the left side of the DNA strand? Okay, let's see how you did. Awesome. I hope you guys have gotten the hang of this. GCAT's pretty easy to work with, but we will be seeing it throughout the entire unit. So to practice what we've learned today, on the right side of your paper, I'd like you to make a column of mRNA. So we're taking this DNA in the center and rewriting it, transcribing it as RNA, mRNA specifically. This is called transcription. And because we're creating RNA, this now follows the GCAU rule. So I want you guys to try and turn that center, that center DNA molecule into an RNA molecule on the right-hand side of your paper. Okay, let's see how you did. So the first base in that DNA molecule is adenine, A. So A in our GCal pairs with U on RNA, so that was pretty easy. We know that C and G always go together there, so that one's not too bad. The only tricky part with this is when you look at DNA, that third base, that T, there's no T in RNA. It's a U, but T and U don't go together. T and A go together. There are A's in RNA. If you look at GCal, where you would want to put T, A is still there. So A's are allowed in RNA. Basically, guys, the only thing you have to remember with this GCal rule, it's just like GCat, but anytime you would want to write a T, you would just write a U. So if you compare your DNA in the left column, see how it starts with TGA, and you compare that with your RNA in the far right column, it's UGA. They're exactly the same, except where I have a T, I make it a U. That's it. So replication and transcription are very, very similar, but the biggest difference is instead of writing T, you write U. That's it. So I have some more practice for you, so you're free to use your notes. I don't mind if you work with a partner, but you're going to be quiet while you do it. The partner is so that you can try it, the partner can try it, and then you can kind of compare answers. So if you do get stuck or lost, you have someone there that might be able to help you out. Okay, so we're going to review this tomorrow before moving on as well. Okay, guys, see you tomorrow.